So I'm going to share a couple, themes, this morning, and really going to take a little more of a technology, view and shift. I think really the first key message is that I think we all have to realize we're very much in the early phases of, technology development when it comes to the genome editing space. And internally we've been working in the genome editing space, for 5+ years. But I would say over the last 5 years, we've just seen a rapid pace of evolution and really creating new opportunities to drive applications and approaches for the technology. And so I think as we talk about the technology and I think how we see the technology being used today, I think we all also have to realize that probably tomorrow what we can do with the technology will be different. Internally, we've spent again the last 5 years, looking at Cas9 nucleases, trying to understand the diversity of nucleases. We've really worked hard to think about applications of Cas9, from an architecture standpoint, and really understanding how to characterize the use of the Cas9 nucleases to really ensure broad application across the crop genomes that we're working with. And that gets to really understanding sort of PAM motifs and really how readily we can deploy the technologies. And more recently, you could see that the innovation's continuing. Internally, we've identified some miniature nucleases, which really has, I think, some positive impacts on sort of payload and sort of ease of use. You've seen transitions to next generations of base editing technologies, and more recently with prime editing, you're really integrating a number of molecular tools, such as the nickase cutting with nucleases, and sort of partnering up with reverse transcriptase technologies to really drive a more targeted base editing approach. And so I think today we're in a great spot. It's an exciting place to be. But I think as we look to the future, that the opportunity is going to continue to evolve, and we're just going to see, I think, more robust applications come forward for us in the agricultural sector. As we think about the technology from a breeding tool standpoint, we've certainly seen opportunities for CRISPR to really drive improvements in our breeding strategy, sort of open up the opportunity to drive improvements in our breeding strategy. And I really love the idea, sort of inspired by nature, because in very similar ways, we're learning, from our breeding organization, how to better leverage, the tools to drive, greater variability. On the bottom left-hand side there, just an example of how we are trying to leverage, CRISPR, technology to drive improvements in traits such as disease resistance. And today, breeders are a bit constrained in sort of how they access, I would say disease resistance. They very rarely, move outside their breeding pool because of the challenges associated with doing that. And I think CRISPR, creates an opportunity for us to really select alleles, from, progenitor, species, teosinte, moving tropical, traits into temperate, germplasm in a more readily fashion. And I think what that does is it really allows us to more readily respond to grower needs and shifts in a disease pressure, and to do that and deliver products that are superior to what we could have traditionally delivered with a breeding process that takes a little bit longer and tends to have challenges with, bringing along some unintended, donor genetics. It's really hard to clean up and really disrupts that yield scenario that the breeders have created with a specific inbred. And we're also seeing opportunities in the breeder organization where, as our genomic tools become more readily available, we get deeper genomics knowledge, and we're seeing areas of the chromosome where we basically identified inversions from a chromosomal standpoint, and that really limits the ability for breeders to select recombination within that within that region. And in the case example on the right hand side there, we have identified regions on a chromosome 2 where there are some significant QTLs or significant traits associated with those regions that the breeders just don't have access to in a selected number of our genetics, because they've basically they're working through an inversion issue, and we've limited recombination. Using CRISPR, we've been able to sort of correct that. We've basically been able to, fix that inversion. And what that does is it really opens up the breeding organization, to identifying and selecting for those traits, in that particular region of the chromosome. And we've demonstrated we can do this. This has been a very challenging process, but we've demonstrated we can correct that inversion. And we're in the process of understanding, how we're driving an improved recombination, in this particular area.
5 minutes.
And so again, just an exciting opportunity. Just really two key examples of how we're starting to see, the CRISPR technology really enable and improve our overall breeding strategy. Very much like we heard a little bit earlier, there's this opportunity for capability building and sort of building local infrastructure. And while this is not a perfect process, we have really worked hard to engage NGOs and really targeting, I think, segments of agriculture that really are not core to our business today, but really have a need from a technology development standpoint. And I really highlight 3 examples where we're partnering with NGOs, Summit, ICRISAT, and even the Danforth Center, where we're trying to work on building, technology competency and capabilities, and really enabling, third parties to not only access the technology, but to train, scientists to deliver the technology and really drive local solutions that are really important for a number of, small shareholder farmers, and really trying to address local needs and some, challenges from a climate standpoint. And maize lethal necrosis in Africa is a great example, as well as, looking at some of the challenges associated with, food security and sorghum and millet. Just, I think, really good examples where moving the technology into the hands of those individuals can drive local solutions and trying to build training and infrastructure. And very much like Art talked about at the beginning, and we've heard a number of examples, I think, our challenge going forward, and really, I think the biggest challenge for us to fully realize this technology is really the regulatory framework and the social acceptance. And my hope is at some stage we'll get to a regulatory framework that enables the broad use of this technology. Otherwise, I think we'll all be limited and just really seeing the true benefits of this technology, going forward. And I think I don't have necessarily the perfect solution here, but I think it is a great topic of discussion, and really trying to get a sense of how can we do a better job with this next technology innovation in genome editing to build a more acceptable science-based harmonized regulatory framework that really puts the tool in the hands of those who need it and those who can use it, and really doesn't limit it to a limited number of organizations who can fund the journey from a regulatory basis going forward. And so to me, as I look at the technology, I think this is going to be one of our biggest challenges going forward, is really building that policy, building access, and building acceptance from a social standpoint going forward. I look forward to the discussions and thinking about how we could do a better job of this, as a community, to derive the technology use and acceptance.